First, I want to thank the network for supporting my research and inviting me to join this meeting, share our work. For the audience here, there's no need for me to explain what stress is, but I do want to point out that stress, by definition, is much more broader than the stress stimuli. Most times, it's genotoxic stress that we introduced into the experimental system, whether it's cell or animal model. And it includes external, internal, and what we care more here, psychosocial factors by themselves cause minimal physiological damage by themselves, but epidemiological observation and clinical studies strongly suggest that chronic stress greatly increase the risk for many different types of diseases. And with the interest of our lab mainly in cancer, and here I was showing the example exactly what Don showed you. From a analysis for over 200 of studies clearly showing that chronic stress increase the cancer incidence as well as the cancer survival. While this type of association studies strongly suggest that chronic stress play an important role in promoting the tumorigenesis, mouse work by employing chronic restraint stress, social stress, as we heard many of the presentations before me. And oftentimes for the early work, combined xenograft tumor models having shown that chronic stress can promote tumor growth, metastasis, angiogenesis, etc., and is associated with short survival time in mice. So we think that's easy for us to adapt and consistent with the previous finding that giving periodic restraint chronically before and after introducing human tumor cell line using the xenograft tumor model, we do see this model can cause stress with increased serum corticosteroid level, and at the end of the treatment, you do see the increased tumor volume, accelerated tumor growth. And this type of work indeed provides evidence that chronic stress can promote tumor growth, tumorigenesis, but the underlying molecular mechanism is much less well understood and studied, especially for the signaling pathways that are important and involved in tumorigenesis. So here I want to introduce you an important signaling, the p53 tumor suppressor signaling, which is a critical tumor suppressor. Its importance is well reflected by the fact that this is one of the most frequently mutated genes in all types of the human tumors. So let me briefly introduce you how this signaling works. The p53 itself protein under normal or non-stressed condition is at very low level with very short protein half-life, mainly through the tight negative regulation by MDM2. MDM2 and E3 ubiquitin ligase to promote p53 proteinome degradation. Therefore, its protein level is very low with very short protein half-life, but that can ensure this protein to be alert and sensing a lot of stress signal, mostly genotoxic stress signals, inside or outside of cells. Once it senses that, oftentimes through mediators to modify p53 through phosphorylation, etc., so it can escape the negative regulation from MDM2. Now the protein half-life can increase and the protein level can rise rapidly. And what it does is p53 can function as a transcription factor to transcriptionally regulate many of its target genes, and therefore it can exert its tumor suppressive function. And with the goal to achieve is ensure replication fidelity, maintain the genome integrity and genomic stability by eliminating or preventing the existence of those precancerous cells. So it has been well established if there is loss of p53 or even attenuation of this signaling pathway will impact greatly upon tumorigenesis. And what I will tell you is that our recent work actually shows that chronic stress will have a profound impact upon the signaling to downregulate p53 protein level and function. Let me walk you through our finding as summarized in this cartoon. What we find is chronic stress can lead to the prolonged elevation of glucocorticoids. So it's away from the beta-adrenergic signaling that here studied a lot. And it's through binding to its receptor, which will then go to nucleus, function as a transcription factor. And here what we discover is that it can induce one of its target gene, FGK1, which is a kinase. What it does is this kinase can phosphorylate MDM2, which I already explained to you, a key negative regulator of p53, and this phosphorylation can activate MDM2, increase its activity. So what happen will be increased ability of MDM2 to degrade p53, therefore p53 protein level will reduce. Its function, including apoptosis, will reduce. And in a mouse model that we used using a p53 plus minus mice with only one allele of wild-type p53, so those mice will be more susceptible to genotoxic stress-induced tumor. If you're giving those mice sublethal irradiation, we use four-gray, those mice will rapidly develop thymic lymphoma. And we observe that chronic stress will promote IR-induced thymic lymphoma formation. So that's what we found here. But as we reasoned that chronic stress causes elevation of glucocorticoid is systematic, and the existence of this receptor in many different types of the tissue, this phenotype ought to be more general, and in different kinds of tumor model we can examine. So we want to try, instead of using this really more artificial experimental mouse system, in another model, and here we move into the colorectal cancer, as listed here, with many multi-step events, as well as the development of the tumor with alteration of several important signaling, including p53 here. So we want to ask the question first, whether chronic stress can promote the tumor growth and development of colorectal cancer, and whether the attenuation of p53 function also play a role there. So I don't have data present here, but the first thing we did was measure the p53 level and function in response to stress, and we do see the reduced p53 activity there. And we then go back into the xenograft tumor model. This time we use a pair of human colorectal cancer cell line, HCT116, and they're isogenic with or without p53. As you can see here, loss of p53 can promote tumor growth. Very clear. Now if we apply mice with this pair of cells with chronic stress, what will happen? As you can see that for the tumors with wild-type p53, the promoting effect from chronic stress was very dramatic, and that effect was much reduced when there's no p53 to begin with, but you still can see that chronic stress can promote the tumor growth. So this set of experiments show that chronic restraint and chronic stress can promote the colorectal cancer tumor growth, and attenuation of p53 contributes to this effect. And then we move into an APC mean mouse model, and I told Nathan that I will have a picture to show you exactly the shape, how this type of tumor will look like. So this is a mice containing a allele of mutant APC gene, and those mice will be prone to develop spontaneous intestinal tumor at the age around 14 to 16 weeks, as shown here. And what we found is that chronic restraint indeed can significantly increase the number as well as the volume of the tumors of these mice. And as you can imagine, those mice itself contains wild-type p53. So we want to study the impact from p53 by crossing them with p53 null mice. Now we want to take a look what will happen by giving chronic restraint. You can see by losing p53 already can have quite strong effect on the tumor development and the chronic restraint will have some effect, but was much reduced. And here I want to point out, it's a little bit different with experimental setting. Lisa was showing is that we were measuring this not at the end of their survival, but rather at the same time we sacrifice mice, and then we can more quantitatively comparing that. Well, this is the end point, but then, as I already told you about the function of p53 in maintaining the genomic stability, replication fidelity, then the question come upon is that when you're under chronic stress, even before the tumor formed, the damping down of p53 may have important function to reduce its ability to ensure the surveillance of the genome integrity and everything. So therefore, there's possibility the chronic stress simply through this mechanism as one of its potential mechanisms can increase the DNA damage accumulation and therefore mutation events being increased, and therefore can even have impact as a tumor initiation. So I think that's also one of the question network has been very interested. So we have this system and we can take a look. So as we did, was measuring two quite common type of DNA damage from endogenous DNA damage. One is called AP site, the other is 8-oxo-DG. By analyzing those DNA damage level in normal and tumor tissues collected from the APC mice with or without chronic stress. And as you can appreciate the very clear difference, when there is chronic stress, the increased level of DNA damage can be observed by measuring both damage type AP as well as 8-oxo-DG. And this effect, if we measure the same thing in p53 null background with APC mean mice, then you will see the promoting effect was much reduced. So again, first, support our hypothesis that chronic stress will promote the DNA damage level, and I want to point out that's not only in tumor but also in normal tissue, and the damping down of p53 really contributes to this important effect. And in addition to that, we also measure another important parameter which will sense often like DNA double-strand break, and this is phosphor gamma H2AX will sense and get phosphorylated, and this is one of the early step event occurred when there is double-strand DNA damage. Again, as you can see in both normal and tumor tissue, when you look in the mice under p53 wild-type background, the chronic restraint stress can clearly increase the level of phosphor gamma H2AX, and not so much when you analyze same thing in a p53 null background. So the next step we want to ask is whether that will eventually translate into different load of mutation events in tumors we first studied. So what we did was doing exome sequencing to sequencing the whole genome exome of the mouse tumor as well as normal tissue collected from those mice with or without chronic restraint. So as you can see here, indeed, we observed in the mice with chronic restraint, those tumor accumulated more mutation events compared with the tumors we collected from the mice without chronic restraint. And this difference was much reduced when we measured that in p53 null mice, the tumors from p53 null mice. And we also characterized the mutation spectrum, and we found the majority of mutation occurred as a G residue, which you can imagine with the type of the DNA damage. And the other thing that we find, chronic restraint did not significantly change the mutation spectrum. And we did push one step further, although I don't have the conclusive answer yet. That is, as you can see now, we are comparing the mutation event and accumulation in tumors. And if our hypothesis is true, and if we can find a methodology sensitive enough, whether we can even pick up the mutation accumulation in normal tissue from intestinal tissue collected from APC mice with or without restraint. And we did that by comparing the mutation variation between the normal tissues when we compare with chronic restraint mice versus normal, the control group. But what we found was that quite surprisingly, we will think they are all in breaststring. They should be all identical, but turn out to be there's still quite significant variation among each individual of the mice. We only learned that after we did the exome sequencing, most likely due to the recombination homologue occurred in germ cells. Therefore, it is very difficult to pick up any meaningful difference occurred during chronic restraint. So we actually then change our approach. Instead of directly comparing two mice, which by inheritance they will have difference, we collected two regions of normal tissue within same mouse, with or without chronic restraint. Now we ask the question whether we will see more variation within one mouse at two regions in those with chronic restraint compared with the control group. And while we only have really limited number of the samples, so I think the result is not yet conclusive, but I do want to share with you, we do observe the trend that indeed we do observe a relatively high variation, meaning the somatic acquired mutation in the group, in the normal tissue, in the restraint group, compared with the control group, which again support our hypothesis. So I want to summarize what I told you, but definitely this is something we will continue in our future study. So what I want to summarize what I told you is that we find chronic stress actually can also work through the elevation of glucocorticoids for the prolonged period, then through the GR signaling, through this kinase SGK1 and MDM2-PC3 signaling. One of its particular paths through reduced ability for damage surveillance will cause the accumulation of DNA damage, even before the tumor formation, and that will lead to the mutation events accumulated at a fast speed, and that will contribute to the accelerated colorectal tumorigenesis. So with that, I want to thank all the people in the lab who contribute to this work, and this is in close collaboration with Dr. Fen's group. And I also want to thank Arnie, who is my mentor, and as some of you may know, I told how I got interested into this field largely because of him and his vision, as well as a connection with Paige and everything. And Dr. Chan's group helped with exome sequencing analysis, and the exome sequencing work was performed at Rutgers IUCDI place. And last but not least, I really want to thank the support, including the funding, and from Paige and from the network for our research. And I'll be happy to take questions.